Small fragments of some complement proteins can initiate a local inflammatory response. The small complement fragments C3a, C4a, and C5a act on specific receptors to produce local inflammatory responses. When produced in large amounts or injected systemically, they induce a generalized circulatory collapse, producing a shock-like syndrome similar to that seen in a systemic allergic reaction involving IgE antibodies. Such a reaction is termed anaphylactic shock, and these small fragments of complement are therefore often referred to as anaphylatoxins. Of the three, C5a is the most stable and has the highest specific biological activity. All three induce smooth muscle contraction and increase vascular permeability, but C5a and C3a also act on the endothelial cells lining blood vessels to induce adhesion molecules. In addition, C3a and C5a can activate the mast cells that populate submucosal tissues to release mediators such as histamine and TNF-alpha that cause similar effects. The changes induced by C5a and C3a recruit antibody, complement, and phagocytic cells to the site of an infection, and the increased fluid in the tissues hastens the movement of pathogen-bearing, antigen-presenting cells to the local lymph nodes, contributing to the prompt initiation of the adaptive immune response. C5a also acts directly on neutrophils and monocytes to increase their adherence to vessel walls, their migration towards sites of antigen deposition, and their ability to ingest particles, as well as increasing the expression of CR1 and CR3 on the surfaces of these cells. In this way, C5a, and to a smaller extent, C3a and C4a, act in concert with other complement components to hasten the destruction of pathogens by phagocytes. C5a and C3a signal through transmembrane receptors that activate G proteins. Thus, the action of C5a in attracting neutrophils and monocytes is analogous to that of chemokines, which also act via G proteins to control cell migration. And that small fragments of some complement proteins can initiate a local inflammatory response. Have a great day.